So in the previous video, we have learned the anatomy of dicot stem, where we observe the primary structure of a dicot stem. So I hope you are clear with the structures, right? So now we are going to learn about the secondary growth of dicot stem. So what do you mean by secondary growth? So normally secondary growth means now this is a stem, right? This is the diameter. This is the diameter of the stem. So now this diameter is going to increase. So increasing of this diameter we called as the secondary growth, right? How it is going to increase? By producing new cells. Surrounding this, they are going to produce new cells. By those cells, it is going to increase. Okay, so increasing of its diameter, or you can say girth, that is called secondary growth, right? So secondary growth means increasing the diameter of the stems due to the new cells produced by the lateral meristem. So you have already learned about 3 types of meristematic where cell division take place. So in that, lateral meristem only involves in the increasing diameter. So by producing new cells by the lateral meristem, it is going to produce the secondary growth. Okay, right. So this secondary growth can occur in 2 ways, right? It is going to happen in 2 places, right? So one place, see, you can see these are the vascular bundles. So it is going to happen near the vascular bundles, right? Then the second one, this is the cortex part. Yesterday, the previous video we saw, this is the cortex part. So next is the cortex. Okay, so there are 2 changes going to happen in the vascular and the cortex. Due to that, new rings are going to form. Due to formation of new rings, it is going to happen the secondary growth. Okay, is that clear up to this? Okay, right. Now we'll see how it is going to happen in the vascular and how it is going to happen in the cortex. Okay. Right. Now, see, so first I told 2 region. So first region we are going to see, that is vascular region, where it is going to produce a vascular cambium. In this place only it is going to happen. Right. Now see, these are the vascular bundles. Now I will enlarge that like this. So this is the cambium. Inside we have the what? Inside we have the xylem, outside we have the phloem tissue, right? So inside we have the primary xylem and outside we have the primary phloem, right? So now what is going to happen? Now see, this vascular is in ring like this, okay? So you can see it here. I will draw another ring, okay? Now see, all these are like ring. Here, we have already learned here the ring structure. So all these are forming in a ring, right? Now what is going to happen is, in between these, okay, we have learned yesterday, we have the parenchyma cells. So in between these, we have the parenchyma cells. Here also we have the parenchyma cells. Now parenchyma is a permanent tissue, right? Already we have learned that parenchyma is a permanent tissue, right? So here, what is going to happen? In this place, this parenchyma is going to de-differentiate, which means the permanent tissue, parenchyma tissue, is going to convert it into meristematic tissue. Okay, meristematic tissue. So meristematic tissue, where they can produce new cells by dividing, right? So here in between you have the parenchyma. This parenchyma is going to form the meristematic. You call de-differentiation. Okay, that step you call dedifferentiation. Right, now what happens? Due to that, it is going to form a ring like this. It is going to form a ring like this. Surrounding, it is going to form a ring like this. Okay, right. So normally this, we call this cambium as intrafascicular cambium. So the cambium that is forming between the vascular bundle, this we call interfascicular cambium. I think you don't have these terms, but you have to know this. Okay, so now it forms a ring, complete ring, right? Now I'll draw a complete ring. Let's see how it will be. Assume this is a ring, okay? So now how it is going to form? Due to the parenchyma is going to change into meristematic, right? It is going to form a ring-like structure like this, right? So I am highlighting this like this. Okay, right. So now what is going to happen? This vascular cambium is going to divide. So inside they divide and also outside they divide like this. Outside they divide. Like this, outside they divide. And also inside also they divide like this. Right? So now see, wait. So when this is happening, see, you know, this is the primary xylem. When new cells are forming, these are pushed towards inside. So these are pushed towards inside. These are like this. The primary xylem are pushed towards inside like this. Okay. Then the primary phloem, those are pushed outward like this. Like this, they are pushed outward. Right. So now two new layers are formed. So inner layer which is formed, this is the primary xylem. So which is close by the primary xylem is the secondary xylem. Then here this is the primary phloem, and here a new cell is the secondary phloem. Right? Now see, due to the vascular region, now two rings has been formed which increase the diameter of the stem. So this is one part of the secondary growth which is happening inside the vascular region, right? So they have formed a secondary xylem and a secondary phloem. So what has happened? The primary phloem are pushed towards. So now here the parenchyma says, you know, this place we call the pith. Okay, it should be like this. These are completely, it is like this. Okay, this has fully, the primary xylem has come in. Right. So now the pith, they will disappear. No, there will be no pith during the secondary growth because primary xylem push towards inside and phloem outward. So two new rings are formed. So this is the first type of secondary growth that is happen in the dicot stem. Okay, right. So this is about the vascular region division, right? So I told two region it is going to happen. One is vascular region, that is what we have seen. So newly secondary phloem, secondary xylem has been formed, right? New two layers are added. Then next we are going to see the cortex region. So now this is the cortex part. So we have already learned. So this is the cortex part, right? So here, one of these layer, I will mark like this. One of these layer, right? It is going to de-differentiate again. Normally, we know in cortex, we have parenchyma cells. So here, again, the parenchyma cells is going to converted into the meristematic. So parenchyma is a permanent tissue. That permanent tissue here, it is going to again de-differentiate. De-differentiate means that parenchyma is going to become meristematic tissue, right? So now I'll draw that this, I will take this part here, okay? Right. So here now, this de-differentiated, this part we called as the what? That is you called as the coke cambium. This is the part coke cambium. Okay, this is the coke cambium. Right? So here, the cortex, from the cortex, one parenchyma layer is going to de-differentiate into meristematic, and we call that as the coke cambium. So inside this, that is vascular, here coke cambium, right? Now what is going to happen here? It is a meristematic tissue. So they are going to produce new cells up and down. So they are going to produce some new cells upward. That is also parenchyma cells. Okay. And now new cells downward, that is also parenchyma cells. Okay, I hope understood, right? Now this is not this layer. This is the cells that are produced from this. Okay, then I will draw this here. Okay, I erase this. See, I'm going to draw this layer little bigger up here. Okay. Now, when we talk about these they have produced parenchyma cells, and these parenchyma cells, in some of them, they have some depositions like this. I will draw that thick, okay? Like this. Those depositions, we call that deposition suberin. So, suberin has been deposited some of the cells where it will not allow any water or any other molecules through these cells. And there will be some parenchyma cells. Some are suberin deposition, some are parenchyma cells. Right? These suberinized cells we call as what? Those cells only we called as what? Coke. Okay? Coke. So below the coke only we have the coke cambium. So after sometimes what happens? These parenchyma cells which were in the primary, they will be shed off. They will be get rid of the this part. Okay. So here you have the epidermis. So from this it will get rid. Okay. So here you have the coke, and this is the coke cambium part. Right. Hope now it is a little clear. Right. Now we have the parenchyma cells. So here, this layer we call as cortex, but it is newly produced. So we call this as the secondary cortex. This is called secondary cortex. Okay, so first see, from the parenchyma cortex part, one layer is de-differentiated into meristematic. This meristematic tissue is going to form, it's going to divide. So it is going to divide up and down. So it is going to again form parenchyma up, again parenchyma down. So in the parenchyma up, some of them have suberin, some of them are suberinized. So we call them as the coke. And this cambium we call as the coke cambium. And down you have again from the parenchyma, this is the cortex. As it is formed newly, we call them as the secondary cortex. Now see, so altogether coke cambium, and secondary cortex, all of these three, okay, we called as what? We called them as periderm. You have this in the resource book, periderm. Okay, periderm. Okay, periderm. Okay, is that clear up to here? I hope it is clear, okay? Here, two new layers are formed. So one is the coke, the other one is the secondary cortex, right? So now you have an idea. See, first in the vascular, we added 2 new layers, that is secondary xylem and secondary phloem. And here we have added cork and secondary cortex, right? So now you have altogether, we have added 4 layers where it has occurred the growth. Okay, is that clear? So this is about the secondary growth. So I will explain how it has, like, what are the new from the primary to secondary, what are the parts it has come, right? So from the primary, you have the cuticle, then the epidermis, then the cortex, then we had the endodermis, then the pericycle, then the vascular. In the vascular, primary phloem and the vascular cambium and primary xylem. So these are the things we had in the primary structure. Now, after secondary growth, see, you had the cuticle, same. Right, I will write again. Cuticle, epidermis. So in the cortex, you added new layers. One is cork cambium. Then the secondary cortex. It comes under cortex, right? Then you had the endodermis as it is. And here you had the pith here. Right? You had the pith in primary, endodermis. Now you had, in the secondary, you had the primary phloem, then the secondary phloem, then again vascular cambium, then secondary xylem. I'll write here remaining primary xylem. And here you don't have it. So you have added some new layers from here. Okay, is that clear? So this is about the secondary growth. So if you go through the resource book, read thoroughly, then any doubt you can let me know.